Diseases can be caused by a lack of or non-functional protein, such as haemophilia, insufficient amounts of protein synthesized, such as thalassemia, presence of malformed protein, such as Alzheimer's disease and Duchenne muscular dystrophy. Proteins from an external source can cause disease, such as bacterial toxins, e.g. cholera, and prion proteins, e.g. mad cow's disease. Proteins have a diversity of functions. Enzymes are required to catalyze metabolic reactions. For example, with the disease phenylketonuria, there is damage to phenylalanine hydroxylase, and with haemophilia, the blood clotting factors don't work. And these are both enzymes which are vital for evasion of these diseases. Transport and storage are another function of proteins, such as hemoglobin and ferritin. Sickle cell anemia has a problem with the hemoglobin. Also, proteins are used for motion, such as muscle proteins. Duchenne muscular dystrophy has a problem with the dystrophin protein. Communication is another protein function. Receptors for hormones and neurotransmitters are proteins. Non-insulin dependent diabetes can result from non-functioning insulin receptors. Structure is another function of protein. Keratin and collagen are examples of structural proteins. Osteogenesis imperfecta and scurvy are both caused by problems with the collagen. Channels and transporters are also made of protein, and these facilitate movements across membranes. Cystic fibrosis has a damage to the CFTR chlorine minus ion channel. Diversity of protein function also involves regulation, as proteins are involved in cell division, protein synthesis, hormones. Insulin dependent diabetes has a problem with insulin, so regulation isn't working properly there. Immunity also involves proteins such as antibodies for self-recognition. Myasthenia gravis has problems with the antibodies to neurotransmitter receptor. Such a wide range of functions requires proteins of all shapes and sizes. Understanding the role of proteins in human health, the scale of the problem. The genome is the full set of genes in an organism. The human genome has been sequenced, about 30,000 to 40,000 protein-coding genes in the human genome, only about twice as many as in a worm or a fly. The proteome, full set of proteins encoded by the human genome, is not the same as the number of genes. 30,000 genes has more than 200,000 proteins and more than 1 million modified proteins. Amino acids are the building blocks of proteins. All alpha amino acids have a common structure. They have an alpha carbon with 4 bonds to a hydrogen atom, an alpha amino acid, an alpha carboxyl group, and a distinctive R group. There are many different alpha amino acids, but just 20 are commonly found in proteins. Some common ways of categorizing are by size, shape, like aliphatic or aromatic, hydrophobicity, polar or nonpolar, charge, acidic or basic, sulfur containing, cysteine and methionine, for example, and imino. Proline is not an amino acid, it's an imino acid. Classification of amino acids. There are various non-polar aliphatic amino acids. These are glycine, alanine, valine, leucine, methionine, and isoleucine. That's glycine, alanine, valine, leucine, methionine, and isoleucine. Polar uncharged molecules are serine, threonine, cysteine, proline, asparagine, and glutamine. Aromatic amino acids are phenylalanine, tyrosine, and tryptophan. Positively charged amino acids are lysine, arginine, and histidine. Negatively charged amino acids are aspartate and glutamate. Tryptophan is a large amino acid. Glycine is a small amino acid. Isoleucine is aliphatic. Tyrosine is aromatic. Cysteine and methionine contain sulfur. Proline is an amino acid with an I. Amino acids can be part of more than one grouping, e.g., cysteine, cis, is sulfur-containing, uncharged, and polar. Methionine, met, is sulfur-containing, aliphatic, and nonpolar. Phenylalanine is aromatic and nonpolar. Valine is aliphatic and nonpolar.